We have till now we have understood there are three types of cones. Okay, red, green, and blue cones. Remember, color vision is not just like one cone which detects one color. It is actually how strongly each cone will respond to a different wavelength of light. Okay, I'll explain this detail now. See here, what I've drawn here is a light absorption curve. In x-axis, this represents the wavelength of light in nanometers, and that ranges from 400 to 700 nanometers. Now, y-axis represents the light absorption. Okay, and it's a relative absorption, or we can also tell it as sensitivity of photoreceptors. So if you can see here, there's 25 absorption of percent absorption, 50% absorption, 75%, and 100% absorption by the cones. That is what is represented in the y-axis. Now, just observe there are three colored curves. Okay, one is in blue, the other one is in green, the other one is in red. The blue curve, if you observe, it peaks around 445 nanometers. Somewhere around 445 nanometer, the blue cone is peaking. And it is sensitive to shorter wavelengths. Can you see that? It's somewhere very close to 400, not somewhere here, right? 700 and all will be like closer, that will be a longer wavelength light. So this blue cone is responding or sensitive to short wavelength light, and that corresponds to blue light or violet light. If you observe the green cone, the green cone peaks around somewhere around 535 nanometer. The peak of green cone is drawn, and this is sensitive to the medium wavelength, somewhere in the range of maybe 450 to 600 nanometer. Somewhere in the middle of our visual spectrum, green cones are sensitive, and this corresponds to the green light. Okay, the red curve, if you observe, it peaks around somewhere around 570 nanometer, and it is sensitive to long wavelength light, and this corresponds to the red light. What you need to understand here is these curves are broad and overlapping, and they are not narrow. So we have not drawn blue cone, green cone, and red cone, something like this. Okay, what has happened? There is overlapping of blue cone, red cone, and green curves. All the three curves are overlapping, and this is very important for understanding the color perception. Okay, so there is another dotted line clear that represents a rods curve, and the peak of rods is the rods mainly responds around 505 nanometer. Rods are more sensitive in dim light, that's why they are mainly responsible for scotopic vision, and rods do not contribute to color vision. Remember, that's why in dim light, no, everything appears grayish color because cones are not responding. It's only in the dim light only the rods are responding. So there's no single wavelength light activates only one cone. At each particular wavelength, it will at least stimulate two or three cones in different proportions. For example, if you see a yellow light at 580 nanometer, that will stimulate both red and green cones. Okay, see for example, this is a yellow color. I've written Vibgyor, right? So if you see a yellow color, what has happened is it will stimulate both green and red cones strongly. Okay, and brain will interpret this combination as yellow. So the color is the pattern of activity which is happening in the cones. Clear? So this forms the basis of trichromatic theory. That is, the color perception depends on the relative activation of L, M, and S cones. So that's why we tell the trichromatic color vision theory. No, that tells that there are three types of photoreceptors which are present in the retina, and depending on how much each of them, or how each cone, or how what percentage of these cones are stimulated, depending on that, there is the brain will perceive as a different color. Now just this, I'll just take an example here. Okay, at 580 nanometer light, okay, remember the red cones are stimulated up to a 99% of stimulation value, green is 42%, and blue is not at all stimulated. See now, if you go back to this graph here, 580 nanometer somewhere comes here, right? I'll just use a different color. Okay, so this is somewhere here is the 580 nanometer. This is 600, 500 to 600 wavelength. So 580 somewhere falls here. At this stage, what has happened? 99% stimulation of red cones. There is 42%. Yeah, somewhere here the graph is 42% stimulation of green cone, and blue curve, if you see, it is outside. So blue is not at all stimulated. Now the your nervous system or the brain will interpret this set as a ratio of 99 is to 42 is to 0. So your brain will perceive it as orange color. Hope you're understanding again. Remember, at a particular wavelength, I'll just come again. Yeah, just see here. This is 580 nanometer. At 580 nanometer, we are seeing the color orange. How the brain interprets this? Because there is about 42% stimulation of green cone, 99% stimulation of red cone. So brain will usually, this is the, instead of RGB, like easier way to remember is RCB, Royal Challengers Bangalore, right? Instead of C, you make it as G. So this becomes RGB. This is the order in which our brain interprets how much, which our brain will interpret what percentage or what is the stimulus value for these three types of codes. So there is 99% stimulation of red, there is 42% stimulation of green, blue is not at all stimulated. So brain will perceive it as orange color. Some other examples are RGB ratio of 0, 0 and 97% will be interpreted as blue color. Yellow will be 83 is to 83 is to 0. It means what? The red cones have been stimulated up to a stimulus value of 83%. Green cone is also stimulated up to a ratio of 83%. B will be no, nothing, okay, 0. So green will be 31, 67, 36. So we'll just see green, okay. Yeah, so this is a green color in the graph here, absorption curve. Just observe the green color now. There is stimulation of blue, there is stimulation of red, there is stimulation of green. So if you see the ratios, which is the highest, green is the highest, right? Maybe these values are not corresponding on the left side, but remember, maximum green is stimulated, very little blue and red. So you can see here, blue and red are little, but highest green stimulation. So that is when your brain will interpret us as green.